Good evening, ladies and gentlemen. Welcome to Monash University, the Monash Biomedical Imaging Centre. My name's Luan Ismail. I'm the Executive Director of the ICT for Life Sciences Forum, and I'm pleased to welcome you to this evening's presentation, the second we're holding here at Monash University this year. 2013, in many years to come, might go down as one of the critical years in brain research. In April, President Obama announced a $100 million funding initiative from the US government for brain research. I think he called it the Brain Research Through Advancing Innovative Neurotechnologies, initiative that was launched in Washington and funded through the National Institutes of Health, the Defence Advanced Research Program, and the National Science Foundation. In Europe, the European Commission, under its 7th Framework Programme, announced a €1 billion 10-year program to understand the brain. So it's an exciting pair of announcements. Australia has a tremendous reputation and history in neuroscience, and I should also welcome a special guest this evening, Emeritus Professor Derek Denton, who's an internationally recognised neuroscientist and of course the founder of the Florey Institute. So this evening, Professor Gary Egan from Monash University is going to discuss what these two announcements mean, particularly for brain research in Australia, how we might be able to be a part of what's happening globally. Just to introduce Gary. Gary is a National Health and Medical Research Council Principal Research Fellow and the founding director of the Monash Biomedical Imaging Centre here, a research platform that encompasses the biomedical imaging research facilities at Monash University. He's the lead investigator of the Victorian Biomedical Imaging Capability and the deputy director of the Australian National Imaging Facility. Please welcome Professor Gary Egan.
Well, thanks very much, Luan, for that introduction. And I'd also like to welcome you all here to Monash Biomedical Imaging. It's great to have the turnout, and particularly for such a hot evening. I'm really pleased that, as Luan introduced this talk this evening, to be able to talk to you about where we're situated here in Australia, and I guess particularly in Melbourne, around these international large-scale neuroscience brain research initiatives. And the title of my talk, as you can see, New Horizons in Human Brain Imaging in these large-scale international initiatives, is actually sort of half borrowed from a series of meetings that have been held in the Pacific Rim area over the last 5 years, initiated from colleagues at UCLA, Art Toger and John Mazziotta and Jack Van Horn, and a number of my colleagues who are here have participated in these meetings, which when they started were looked at a little bit like a nice reason to go to Hawaii in March, and it still is a good reason to go to Hawaii in March, but it's actually, I think, represented the need for more international coordination, and particularly in the Pacific Rim area, where there's obviously very rapidly growing economies, as well as, of course, the United States and Canada and Australia. Now, my talk this evening is a slight retrospectus, looking backward a little bit, and it's sort of also a little bit of a personal story, because my interest and involvement now in these large-scale international research initiatives derives from actually my PhD work when I was a physics PhD student many years ago. I won't say how many. And so I'm going to tell you a little bit about my experience at that time and how I think in the more sort of immediate recent past and where we are now in terms of these brain initiative and also the Human Brain Project that Luan mentioned, what it means in terms of sort of neuroscience generally, and I guess more specifically what the implications are for brain imaging and neuroimaging. And then I'll tell you a little bit about the immediate future, or at least one possible future that we hope might come to be. So this photograph is from a detector which I worked on in my PhD, which was based when I was at Melbourne University doing my PhD studies. And it was a detector in Geneva, underground about 100 metres. It was constructed, as you can see there, during the 80s, and it was constructed with one specific purpose in mind, which was to try and discover two fundamental particles, the W and Z bosons, which at that time had been theoretically postulated but had not been identified. And I think that's a key point now when we look forward, that these particles, which were actually discovered in 1983 and a few years later were sort of measured to great detail or great accuracy, the masses of these particles, was actually the forerunner of what just last year was announced as the discovery of the Higgs boson, another fundamental particle which was conceived, as it says in this slide, as a mathematical tool by Peter Higgs and colleagues to make the masses of these particles be able to be calculated. And so it was a theoretical based approach to predicting the existence both of the W and Z boson, which were then discovered within about 15 years. But of course the Higgs, which was only announced its discovery last year, took another almost 30 years for the discovery to be made. And through that time actually involved the expansion of the research groups in size and complexity from the 50 or so people who were involved in the UA2 experiment that I worked on up to what is now 3,000 or 4,000 or 5,000 scientists. So that's been a transition in terms of large-scale particle physics research over the last 30 years. And another point of that project was that it was very single-focused. It had the discovery of those particles as its key question, and the actual discovery then led to the awarding of the Nobel Prize in 1984 to Carlo Rubbia and Simon van der Meer for their contributions to those developments. So it's incredibly theoretically motivated science. It's focused with large teams towards complex systems, and then, of course, fundamental discoveries and breakthroughs, which I think is an interesting perspective to take from where we are at in neuroscience today. I'm now going to turn to the parallel science, which I'm now working in neuroscience and neuroimaging. And this is a phrase or a quote from George Santayana, which I'm sure many of you have heard or are aware of. Those who cannot remember the past are condemned to repeat it, or put another way, those who cannot learn from history are destined potentially to repeat it. And I put this up here because we've just heard that there are a number of two international large-scale initiatives underway, but we mustn't forget that just over 2 decades ago there was another large-scale initiative, the Decade of the Brain. Does anyone remember the Decade of the Brain in the 90s, which was at that time touted as being an incredibly important development? In fact, it was announced in not too dissimilar terms as to what we saw earlier this year. It was announced by the President of the United States, and he made a speech in Congress saying a new era of discovery is dawning in brain research. And he had goals, as you see here, to enhance the public awareness of the benefits to be derived from brain research through appropriate programs, ceremonies and activities. So relatively undefined, but certainly incredibly important to have the President define a decade as a period within which there would be concerted effort by presumably the American scientists and international scientists. So that was an exciting period, and I guess the question is, does anyone remember what particularly came out of the Decade of the Brain? Anyone suggest a discovery that came out of that decade of research? There was obviously lots of important breakthroughs, but it's a bit different than what I was putting up early with the W and the Z boson theoretical postulate, the experiment, the discovery, and then the Nobel Prize being awarded for that. There's a different paradigm that occurred during the 90s, but there were some important outcomes, and I've taken these points from a strategic plan document from the National Institutes of Mental Health that Thomas Insel is the director of, and he's now actively involved in the planning for the Brain Initiative. And again, some of the emphasis of the benefits of the Decade of the Brain were fundamental insights into how the brain works, including new ways of visualising the brain with imaging technologies. That's actually one of the most prominent outcomes from the Decade of the Brain's research program. And he used that point in his preamble to now talk about another decade of discovery in the current decade that we're in, that those benefits from the Decade of the Brain have set the scene for a new century, or sorry, a new decade of scientific opportunities, which we couldn't have imagined 10 years ago. So that's an interesting perspective, that Decade of the Brain in the 90s is now, through the NIMH's strategic plan, initiated another decade of brain research. But again, I'd sort of ask the question, does anyone remember a discovery in that decade which sort of is a signature discovery from the United States? Well, I'll put this up because I'm sure some of you are thinking of this. It's a report in the early 90s by 3 different groups of the first functional brain imaging studies that were done with MRI and with functional MRI. So Agawa and Lee showed oxygenation levels of blood. Changes in those oxygenation levels could act as a contrast agent with MRI, and this is now the workhorse of functional imaging. And in fact, it was reported a year or two earlier by a colleague who's an Australian, Keith Thorborn, who was working at Melbourne University at the time, who reported in blood samples changes in the magnetic properties of hemoglobin from the oxygenated to the deoxygenated state. So that was a fundamental discovery, and the translation of that into imaging techniques then occurred very rapidly and led to this famous image, which was on the cover of Science by Jack Beliveau's group at the time. They actually used a contrast agent in that experiment, and others a little bit later did the standard, what we now call BOLD, or blood oxygenation level dependent method of functional MRI. So I think that was an important discovery. That's probably what Tom Insel was referring to in the strategic plan for the NIMH. And in fact, I would say that that discovery contributed towards the announcement and the award of the Nobel Prize in Medicine in 2003 to Paul Lauterbach from the US and Peter Mansfield in the UK. Even though they themselves weren't doing that functional imaging, it was their developments of fast echoplanar imaging in the early 80s, which actually was then taken advantage of by Beliveau and Co. and others. So that's a nice parallel, which I think highlights that fundamental discoveries will, in time, come out in the brain imaging area as well as I was showing earlier, in the particle physics community. So that's a little bit of the past. Where are we at now in the present? Well, again, I'm going to start by showing you the current day instrumentation that is used in particle physics. This is the Atlas experiment, Detector, again in Geneva, underground, about 150 metres. I was there in July this year and had the good fortune to go down and have a look at it, and it's completely awe-inspiring. Like it's just phenomenally unbelievable to stand there and look at this incredible man-made device, which I think has something like 10 to the 11 detector pads within it. So that's about 100,000 million detector elements in it, which actually is not quite as complicated as the human brain, in fact. The human brain has 10 to the 12, or a million neurons, and even more than that, it's got like a million billion connections between those neurons, so 10 to the 15. So the most complicated man-made device is still not as complicated as the human brain, which I think is an interesting parallel. And this detector and another detector there were developed over the previous decade or more, again with a single objective in mind, which was to try and discover the Higgs boson that I mentioned earlier, which came out of this theory of the standard model. And after more than a decade of concerted effort, but certainly an incredibly large team effort towards this, it was announced, as we know, mid last year that the Higgs boson had been discovered, and it was videoconned around the globe with the two detectors presenting, showing some events which highlighted the production of the Higgs boson in there, and then the plot of their data, which I put up here for, I guess, highlighting the incredibly small effect that they were looking for in this distribution of their data points, and then a very small excess bump of events here, which was their identification of a number of events leading towards the discovery and announcement of the Higgs boson. So that was very exciting. I also thought these images from the detector were interesting, but certainly not as impressive as what we can produce with brain imaging, and I'll show you an image at the end which I hope you'll agree with me can definitely do better than the images from the Atlas detectors. And again, as we know from just a month or so ago, the original developers of the mathematical theory, Peter Higgs and Francois Englert, were both awarded the Nobel Prize for their contributions towards the discovery, the theoretical basis led towards the discovery of the Higgs boson. So I think that's very appropriate and amazing result with such a huge amount of people's effort going into the work for those detectors. And in parallel, what's the present situation for us in brain imaging research and perhaps more generally in medical imaging? I've got a few slides here from Vivian Lee, who is from Utah and a senior figure in the medical imaging community, the International Society for Magnetic Resonance in Medicine. And she gave the Lauterbur lecture here at Melbourne last May. And she really sort of, I think, threw a challenge to us in the medical imaging community and brain imaging community to address the role of MRI, and I guess I'm sort of interchanging here MRI and neuroimaging a little bit freely, but she threw a challenge to us to ask what are the contributions that MRI has made in society and towards science over the past couple of decades. We've heard there's been a Nobel Prize in MRI. It's clearly got a massive impact in clinical medicine, but are we using it in the research community in the way that we need to really gain the benefits from this amazing technology? So she acknowledged that we're developing great technologies, we're probably advancing scientific knowledge, but really are we advancing human health? And she highlighted there three more questions, which were fundamentally, are we studying the right problems with these detectors? And are we proving that imaging is the right way forward in MR clinical research? And in particular, are we adding value? And she actually argued that we're not really addressing those 3 points. So that was quite controversial to some extent, but also I think generated a lot of discussion and made people think about what we really need to be doing with imaging, be it in this more MR clinical research environment or particularly in the neuroimaging context that's more relevant tonight. One of the points she made, which I actually heard a lecture earlier today about the review process or the publication process that we all exist in here with Publish and Perish, is that in imaging journals, as you can see there, the median number of subjects involved in a study is only 32. So there's quite a big range from 6 up to about 1,722 subjects. But the average number of subjects involved in an published imaging experiment is only 32, which is relatively low, and compared certainly to other journals in cardiology and medical across the board, there's more than 200 subjects, human volunteers or patients involved in the published work. And so she highlighted this really comes down to the key problem of the reliability of the findings which are being published in journals. And the lack of studies which try to reproduce published studies is another limitation. And it really highlights in her suggestions for improving the quality of this work is to develop multi-centre, and I've put here in brackets, international consortia to try and bring together data sets which address the same question, need to be able to be brought together and merged into a single data set, but to gain sufficient statistical power to be able to really address very rigorously and with high reliability questions that are based on imaging. And another point she made, which I've just listed here for reference later, is that she was arguing that we need a strong workforce skilled at comparative effectiveness, and in my context here I put interdisciplinary, which really highlights the need that imaging on its own is unable to appropriately and adequately take advantage of the expertise or the opportunities that the techniques offers. So these were important statements from Vivian Lee, and it's now been followed up with a scientific paper published in Nature Reviews Neuroscience just a few months ago, which has underscored the same problem, and it's the failure of having sufficient or adequate statistical power in experiments. So why small sample size undermines the reliability of neuroscience. So it's the same issue that Vivian was highlighting, but actually now in the context of neuroscience, both from wet lab through to human clinical research. And their assessment of a huge number of published neuroscience studies was that they estimated the median statistical power in neuroscience is 21%. So for anybody who's involved in research where you would ideally have statistical power of 80% or higher, and in fact today earlier I heard advocated that we should be doing studies with a statistical power of 90% or higher. This is just, it's really rubbish. It's just inadequate, and it means that we're not really thinking adequately about how to tackle the neuroscience problems. So that's a pretty strong criticism, but I think we need to listen to that, and I think a lot of people are sitting up and taking notice of this issue. Although not everybody is bedeviled by that low power, because I'd now like to tell you briefly about a current project funded by the National Institutes of Health, which is the Human Connectome project. Some of you may have heard of that. I know some people here are using data from the Human Connectome project. And it's a $50 million project over 5 years. It's certainly a large-scale project, but it's not really a breadth to the same level as I was explaining with the particle physics experiments. It's still a group of 3 or 4 universities and 30 or 40 investigators, as you'll see. But it's tackling the crux of the problems that I've just really identified. It's an effort by NIH to comprehensively map neuronal connections and variability in healthy adults on the left-hand side here at the macro scale, the macro connectome, rather than the micro connectome, which other researchers have been developing and publishing. It's a large study of over 1,200 healthy adults and 300 twin pairs and their non-twin siblings. It's using MRI comprehensively, so it's imaging, it's taking advantage of what was advocated from the decade of the brain. The discoveries that imaging provides this phenomenal opportunity for non-invasive analysis and an understanding of the brain, both structurally with the DTI tractography techniques and also functionally with functional MRI. And there's also 100 subjects who are being examined with MEG and EEG. So it's a very large experiment, large data sets, comprehensive behavioural testing, and also blood sampling for genotyping. That's the first list of project goals. And the second list here is to make the data freely available and to make it available as it's basically coming off the scanners. And I thought that's a really interesting change of scientific social interaction, that people now are actually collecting data sets, making them available on the web or by other means as soon as they're available, even before any publications have necessarily been made by the chief investigators of this project. And they've also been funded to develop informatics platforms to help in actually analysing the data and doing discovery science using that data. And their particular emphasis is to relate the connectivity measures with structural, particularly structural and functional, with heritability using the genetic information. And they see this as a sort of a platform for expanding into future areas of brain disorders. And I think the success to date would indicate that that's probably a likely occurrence, given that they've been incredibly productive and have actually adhered to what they've said they'll do. They'll share their data with us as it comes off the scanner. You can actually order the data sets online as a connectome in a box. Has anyone here ordered a connectome? I know a few people have downloaded data sets, but it's possible to just, I think it's $200 for the cost of the disk, and they'll even recycle your disk for the next time round when you want the updated version. So you can order the connectome on a few terabyte disk. As they point out here, it's a very large data set. To put it mildly, it's enormous. They're up to 6 terabytes now for 256 subjects. So you do probably need to put an order in rather than just try and download it through the internet. And the reason I've highlighted this experiment is that it's actually changed the paradigm, the way we think about doing science now in brain imaging, and I think a little bit more generally in neuroscience. It's one of the high-profile NIH large-funded projects. It's a 5-year project. It's primarily North American, but it also has Oxford University as a key collaborator. And they've sort of reversed the engine here. They're producing large data sets, a bit like the particle physics experimentalists do, and then they're putting it out into the community, and it's sort of like reversing the mechanism which has sort of been established in the high-energy physics world. And I think it's actually going to become mandatory for imaging and other neuroscience researchers to be able to make their data accessible by other researchers within a very short period of time after having collected it. And by short period of time, I really mean within 12 months at most, and potentially within 6 months. And so again, that's changing the way that we'll need to be able to do our research, collect our data and get it analysed, more in real time rather than sort of after the fact. So that's a little bit of the recent past, and that Human Connectome project, as I say, I think it's finished in 2015, so it's been running now for three years or so. But the more immediate now, as Luan mentioned, has been the establishment of the Human Brain Project in Europe and the Brain Initiative in the United States. So I'm not sure if many people are familiar with the scope of the Human Brain Project. In fact, some people call it the Human Brain Project, others call it the European Human Brain Project. Depends which side of the Atlantic you're on. I know the Europeans would like it to become a national project in the same way as the particle physics projects have become international. And so they're very open towards participation from groups in Asia Pacific and of course the US. It is a 10-year project and it has very broad and ambitious goals to understand the human brain, enabling advances in neuroscience, medicine and future computing. And this point about future computing is actually critical because this project has not been funded out of the Biomedical Sciences Research Program or a neuroscience initiative per se. It's actually been funded from a Future Emerging Technologies program in the European Union under the FP7 platform. And in fact, over half of the funding, which as you can see down the bottom here, is 1.2 billion euros over that 10-year period. Over half of that funding will actually go into computational facilities, high-performance computing facilities, neuromorphic computing technology developments, and technology software platforms to utilise and provide access to those computers. So it actually has a flavour, to my mind, like what we were seeing earlier with the experimental particle physics collaborations and sizable investments. And this is actually headquartered in Lausanne, which is just a little train ride up the lake from Geneva. So the Swiss are thinking very strategically. They brought together 256 researchers, primarily in Europe, but also a few from the US, Japan and China, And as I say, it's actually started 1st of October this year. It's actually on a 2.5-year ramp-up phase initially to be able to determine by review whether or not the EU will then continue the funding for the further 7.5 years. So it's in a ramp-up phase, but it's obviously an exciting phase, and it has these goals, which I won't go through in detail, but suffice to say, it pretty much covers everything you could think of. If you have a look in the report that was put to the EU, it includes really all scales from the genomic level right through to whole brain systems. It includes animal models, particularly mammalian brain, mouse models, human studies. And as I said, it has a very big investment into the area of computing and future computing to actually learn from the brain how to build the supercomputers of tomorrow. So that's the neuromorphic computing program, which I think is really very novel, and obviously why the Future Emerging Technologies program in the EU chose this to support. So a couple of the initiatives, I've just sort of put the details up here for your interest, to look over a 10-year period at how to build from looking at volumes and neuron counts in human brain regions right up through to the proteome of human cells, including transcriptomes and morphology of neurons. So they're looking at scale of structure across these scales from the macro through to the micro. They also have a focus on looking at the function of the brain across many different common human functional capacities, for example, language and genomic variability of language skills, through to object recognition, navigation, up here into decision-making processes and learning, and then at a higher level, presumably the 10-year point, looking at research programs which will involve understanding, sense of self and presumably consciousness within that scope of research program. So that's an enormously broad scope, very exciting and ambitious, and they have a sort of a similar pathway, if you like, or roadmap for the developments related to the computing platforms, and in particular an emphasis on simulation. So if you visit the Blue Brain project in Lausanne, which was sort of the progenitor to this human brain project, actually simulate neuronal and dendritic architectures and synaptic spines and synaptic contacts from first principles. And it's a novel approach, which I think still has to be proven to be worthwhile, but it's a part of the basis of the development of the high-performance computing platform, with an emphasis on the exascale level all the way through this. And similar to the emphasis in the Human Connectome project, they have an emphasis on developing tools for neuroscience researchers to be able to access the data sets that they'll collect, analyse these data sets, and participate in a global endeavour coordinated through the Human Brain project. So I think there is a few parallels coming through in these projects, and the scale of this now over 10 years I think will completely change the landscape that we as neuroscientists and neuroimaging researchers will be working in. So change gear a little bit across the Atlantic and look at what the Americans have done in response to this. You may have seen this cartoon in the New Yorker earlier this year, the fact that the Americans are not going to let the Europeans take the Human Brain project and run with it on their own. And so the initiative that's been established there actually started the year earlier with this paper published in Neuron by Elvis Sartis and colleagues, which proposed an initiative in the United States essentially as a counterbalance to what the Europeans were doing, but with an emphasis on measuring all of the neural activity within the mammalian brain, so in a mouse model, looking at identifying all of the synaptic activity by virtue of new technologies which people couldn't actually describe or clearly articulate, but which, harking back to the decade of the brain and the discovery and development of new technologies there, led through to fundamental advances that we now take for granted. So the initial proposal was actually called a brain activity map, which was focused on reconstructing a full record of neural activity across complete neural circuits. So looking at all of the synaptic activity that occurs in a mouse brain and actually trying to measure that in real time, which again was put forward as an invaluable step towards understanding fundamental and pathological brain processes. So the good news is that the President bought that story. He was on the front foot. As Luan said, he made an announcement in February of this year, which was published very widely, referred back to the Decade of the Brain, the successes of that, and actually highlighted, as I've circled here, that in the other major project of the 90s, the Human Genome Project, every dollar invested returned over $140 to the economy. So the argument actually has moved into an economic argument and a workforce argument, or the creation of jobs, as is specified in this report of President Obama's announcement. So that was really exciting, but no one actually knew what was going to be done. So there was $100 million announced, and there's actually been more since. But there was no details about what the project was, and it wasn't like some of the earlier experiments that I've described to you, and even the Human Connectome project, which was a very clearly specified project with endpoints and a strategy. And so this has created a huge amount of engagement by the neuroscience community in the US. And just a couple of months ago, there was this interim report released from the National Institutes of Health, which is the working group, which has now in the last 6 months or so worked through all the possibilities of how this funding could be invested and what really is the best way to utilise this initial announcement by the President. It's thought that that is probably the first year investment in the Brain Initiative, and that the similar sorts of investments would occur on an annual basis over a 10-year period. So on that basis, it's sort of $1 billion US into this initiative, but there's actually only 100 million committed new money, and it's not even clear how new that money is. But nevertheless, the working group have now come up with a more concrete list of endeavours that the Brain Initiative will pursue, and a few of them are sort of, I think, quite obvious and expected. Use appropriate experimental systems and models, so many will be actually in animal models. That it will be an interdisciplinary collaboration that will be needed to make these breakthroughs. The expectation is that there's very unlikely that a single researcher will crack the brain's code. That this actually needs to be working both across spatial and temporal scales. I think that's fair enough. And here's another point again about the importance of establishing platforms for sharing the data which comes out of the investments that the Brain Initiative make, and in particular the emphasis on user accessibility. So I think that's a common theme and an important theme. And it particularly should look at disseminating new technologies that come out through the initiative. There's a final point, which again is in the Human Brain Project, is that this shouldn't be just done in isolation from society. It's incredibly important that the ethical and philosophical implications of the research outputs are communicated very proactively with society, and that there is a very concerted effort and money put into making the findings accessible and communicated to the public. So this is a responsibility that we all have as scientists, and particularly with these sorts of investments being considered and made. So that was in September, and just last month, or in fact in October, there was a response by a group of scientists in the US, or primarily in the US. You can see them listed here, who I guess highlighted the challenge of connecting the dots between these different points that have been outlined by the NIH. And they were much more sanguine about what the possibilities were, and they've listed, I guess, the sort of obvious ones against technologies both on and beyond the horizon, so investments in technologies that no one can actually conceive of at the moment, and pointing out that again 5 years ago some of the optogenetics technologies didn't exist, and so the actual pace of change is probably increasing, and it's very important that there be a significant emphasis on that, but not just at the single neuronal activity level recording, so at multiple levels of technologies for macro investigations as well as micro or nano investigations. They actually highlight the need and a real gap in the community for a more theoretical basis to the research that's being pursued. And at the start of my talk, you would have seen I highlighted the mathematical theoretical basis for predicting the existence of a particle, and then the experiments conducted to test for that or identify the mass of those particles. And I think in this approach, there's a very strong emphasis on having theoretical approaches that are much more rigorous to tie together measurements across the spatial and temporal scales. That the approach should also look at understanding brain function in health and disease. They ask that question, and I think they come out on the affirmative there. And they conclude that these coordinated programs really needed both technologically, experimentally, and theoretically to understand the meso and the macroscopic levels of brain organisation. Now, some of these people obviously have, you could say, vested interests. They're working in these areas, and so they are arguing that the areas they're working in should be supported through the Brain Initiative. But as you would have seen in the list, they're very eminent neuroscientists, particularly in the US, and so I think this will probably have a lot of weight into the final decisions about where the first year of brain funding goes. And in fact, there's going to be a request for applications made in January, because the funding has to be actually allocated and committed by the middle of 2014. So it's getting very close to the point where there'll be some allocations made. So that's the sort of where we are now. We've got two exciting international initiatives underway. And I guess to me it sort of highlighted some emerging issues in the neuroimaging community in relation to these two projects, and then exactly how we address these or how we respond from an Australian perspective. And I guess the questions, you can break them up into one around science and technology and another around the sociology of how we do our science. And in particular, what must we do to make the big discoveries? Do we have the necessary technology? And do we need to just focus on working in large-scale, internationally coordinated efforts? Or should we do things differently in Australia and in our research labs here in Melbourne? And furthermore, is this area of research so scientifically heterogeneous that it's impossible to think that you could try and organise so many people on single initiatives, as I was pointing out in the beginning with the particle physics work? And then if we do that, not all neuroscientists can participate in these initiatives. How will we address that? And finally, how can we ensure that those that don't participate directly are able to access the data and the models and make that part of the ethos as to how we do neuroscience? So we have worked on this over the last, I guess, year, roughly, and we've come up with an Australian proposal that I'd like to share with you tonight. It's still a proposal. We haven't yet heard the outcome from this. It's a Centre of Excellence, an Australian Research Council Centre of Excellence application, and we called this a Centre of Excellence for Integrative Brain Function, with the respective universities you can see along the bottom there, all as participants, with researchers from across the country and a breadth of different neuroscience disciplines. And we were, I guess, when we came together and discussed what we could do from an Australian perspective, this was really what we could see as the challenge for us, is to, again, bridge across these spatial scales, but also temporal scales, but in particular, how do molecular and cellular neuroscientists work with people at the brain systems level and with individuals in a sort of circuits and networks level. How can we integrate the research findings and experimental approaches that are made across these three spatial scales, and fundamentally to how to tackle the problem of how the brain interacts with the world? So we advocated that this needs to be multi-scale and multidisciplinary, and it's actually beyond the reach of any single lab, and really therefore behooves a establishment of a centre of excellence. The driver for this is that we now have technologies which can actually provide sensory input to the brain. We can now stimulate through the fantastic discoveries and research done by the Bionic Ear people over the last 30 or 40 years, and there's now over 200,000 people with cochlear implants. So we now know that you can stimulate into the central nervous system, we can get sensory information in. Last year was the first bionic eye that was, which was surgically implanted, and we now have reports of visual stimulation. So that's a very exciting future, and there's two major projects happening here in Melbourne which are working hard towards that end. But what we don't know is how the brain then processes and integrates this sensory information. And so our question was to try and bring together individuals, scientists who were working at the cellular synaptic level together with those at a whole brain systems level, and try and understand what the pathways and the mechanisms are that integrate sensory information. And it's not just a methodological approach, we actually looked at functions which I guess depend very fundamentally on integration of input information. And the three functions that we've chosen and focused on are attention, prediction and decision making. And you might remember I pointed out in the Human Brain Project the series of human brain function work program over 10 years, which also included these prediction and decision as two of the areas of research. So these are functions that are essential for everyday life, such as when you're crossing a road, you have to obviously attend to any stimuli in your environment. You have to be able to predict whether that vehicle will come quickly or slowly, and then make a decision about whether you'll move across in front of that vehicle or wait until it's passed. We all know these three functions are absolutely critical. And we've developed a program of research which bridges these scales of doing functional imaging in humans and primates, non-human primate models, as well as imaging studies and electrophysiological studies in animal models, specifically focused on questions of attention, and having theories which colleagues, particularly Jason Mattingly in the University of Queensland, who's an expert in this area, looking at how attention, for example, alters the gain in low-level neural circuits. So top-down modulation of primary visual input, sensory signals into the primary visual cortex. What are the mechanisms across the whole brain system that enable people to direct attention and to modulate the output from neurons in lower primary and lower visual cortical areas. We similarly have a program in prediction, looking at tasks in humans with imaging, and then studies in rodent models, in fact, predicting the behaviour using theoretical and mathematical approaches of the likelihood and the prediction error to be able to address questions such as which are the brain regions that actually control predictive coding, or in fact which ones are looking at the outliers or exceptions to what our predictions are, because it's the error signals from those tasks or experiences which we don't expect that are really producing the key information for altering our expectations and predictions. And we also have a program proposed in the decision-making science, again with a hierarchical approach and a mathematically based approach to be able to address the questions of which areas of the brain involve on accumulation of evidence, which then tips over into a decision process made by a rodent model in a forced choice alternative experiment, or a human subject lying in a scanner. So we'll see in a few weeks, hopefully, how this proposal has been received by the ARC. Here are the investigators, colleagues from Melbourne University, Sydney University, Australian National University, University of Queensland, and the University of New South Wales, as well as at Monash University, and a really breadth of researchers. So getting people sitting around talking about how we're going to work together was already a significant challenge. But over the course of the year, we've brought that together. And we have a number of international partner investigators listed here, including up here on the top left is Henry Markram, who's the director of the Human Brain Project in Europe, and also David Van Essen, who's the director of the Human Connectome Project in the United States. He's on the proposed board of this centre. So the centre's partners are listed here internationally, as well as a number of other groups. One of the outputs from this is not just understanding basic science and how the brain works, but also the technologies which we hope will go into these number of our associated organisations, the Bionics Institute here in Melbourne, as well as the Small Technologies Cluster and other groups. So we hope that this proposal, if accepted, will have impact both in understanding the mechanisms of integrative brain function, benefit society. We'll have a public outreach program with education and lectures and social media approaches to communicating the findings of this work, and it will also be a focus for collaborations with the international initiatives which I mentioned. And the last thing I'd like to leave with you is just how are we going to actually organise all of this? How does it all come together? Like, it's already very challenging for individual large-scale research groups, but how do they connect between the Europeans and the Americans, between what we do here in Australia, what the Chinese, and there's also a Norwegian brain initiative. And I wanted to point out to you a facility which is based in Stockholm, the International Neuroinformatics Coordinating Facility, which was established about 7 or 8 years ago, and which we joined here in Victoria earlier this year, a joint establishment of a node in Victoria between Monash and Melbourne universities. And at a glance, the INCF basically provides the infrastructure, the tools, the techniques, provides data sets, a place for repositories of data, and infrastructure to help researchers, do neuroscience research, but do it internationally, do it across the globe, share the data that they're collecting, and enable the results to be made available and used by others as quickly as possible. So this is really the purpose of the INCF. As I say, it was set up about 8 years ago, so it was actually ahead of its time. And for the last 6 or 7 years, it's been doing a very good job, but it's suddenly come of age that the INCF now provide this international brokering role, or potentially will do that between these large-scale international initiatives. It currently has these 4 programs in atlasing, in multi-scale modelling, data sharing, and it also has an international congress on once a year. So if you're interested, it's a very nice location next year in Leiden in the Netherlands. The meeting will be held there in August, and we just found out last week actually that meeting will also be held here in Australia in the following year. So the role of the INCF will hopefully become more prominent here in Australia from 2015, when this meeting is held in conjunction with the Australian Neuroscience Society meeting being held up in Cairns at that time, which I think is a really good result. So that's basically where I think we're at the moment. But I do have one last slide, which is about the future and how we actually see into the brain. And you can imagine that with an imaging background, I would advocate that we should use MRI to do this, so no surprises there. But the key point I wanted to make is that we can now do that with a phenomenal new virtual reality or reality immersion visualization system, which has recently been opened here at Monash University. It's called Cave 2. It's a full 360 surround, I think it's 80,000 pixels, or an extremely large number. And what you can see here on this screen, which is in front of these individuals in the room, are actually tracks, white matter tracks in the brain. So from a diffusion tractography data set, this has now been visualised so that you can actually be inside the tracks in the brain in a 360 experience, which I think is really phenomenal. And I'd encourage you to go over and have a visit to the cave if you are interested in getting right inside the brain and looking deep down into white matter tracks. And this is some of the data which has been used. This is from Govinda Pudel, who's here this evening. Thanks, Govinda. I thought I'd finish with your movie, which gives people an amazing insight and experience inside the brain. So thank you for your attention.
Gary, thank you for that comprehensive overview of the international initiatives and what's happening locally. And on behalf of all of us, we hope that you and your submission does get up, and hopefully you can come back next year and tell us a bit about that. I'd like to thank you for joining us this evening and for your support throughout the year. It's been a fairly big year for the forum this year. We've had 11 presentations of this variety, 2 at Monash and the others in the Parkville precinct. Attracted over 1,500 people to those events. The Graham Clark Oration this year, we had 1,600 people to the lecture and 600 people to the dinner. So we've been very busy. So that can only happen with the generous support of our sponsors, and I'm not going to read them all out. You can see who they are there. We're very grateful for their ongoing support and commitment to make events such as this possible. We look forward to coming back in the new year with a continuing program of exciting presentations such as the one we've heard this evening. The last thing for me to do is to wish you a happy and safe vacation, and we'll see you in 2014. Thank you and good evening.